Hello everyone, I'm Yanying Yu, a PhD student in Lars Backer's group in Helmholtz Institute for RNA-based infection research in Germany. Here, I present to you my project, Improve Prediction of Bacterial CRISPRi Guide Efficiency through Data Integration and Automated Machine Learning. In this study, we explore many different aspects of machine learning to improve the model performance and showed how this approach can provide insights in the CRISPR-I system and provide a blueprint for the development of predictive models for other CRISPR technologies in bacteria. As implied in the title, this study focuses on one of many CRISPR technologies, CRISPR-I, in which a catalytically die-cast protein incapable of DNA cleavage, dCAS, is targeted to interfere with transcription of a gene of choice. But why do we need a tool to predict the guide efficiency? First of all, guide efficiency can vary more than tenfold even when targeting the same gene. Second, most existing tools were developed based on the Cas9 system in eukaryotes, and as was also pointed out in a previous study, they also suffer from overfitting. To develop a dedicated tool for CRISPR-I in bacteria, we collected data from CRISPR-I essentiality screens and selected the guide RNAs which target essential genes as training data. In the essentiality screens, the full change of counts of guide RNAs compared to the start time point, which we call depletion scores, shows the effects of the combination of lockdown fitness and guide RNA efficiency. To predict the depletion in a dataset, namely E75 rosette, we applied an automated machine learning tool, AutoCycleLearn. With the help of AutoCycleLearn, we developed models with different feature sets and evaluated the models with the Spearman correlation between the prediction and depletion scores using tenfold cross-validation, which is also applied in the following test. Of the total 574 features, there are mainly two groups, guide features and gene features. Models trained with guide features, such as sequence features, distance features, and thermodynamic features, performed poorly, including gene features such as gene ID, expression level, transcription unit information, boosted the model performance to a Spearman correlation around 0.68. Next, we use G-Sharp to explain the best performing random forest model. Of note, the top features are mostly gene-specific. After we found that auto machine learning can rapidly produce predictive models of CRISPR-I guide depletion, we next asked whether the size of our dataset was limiting the accuracy of our predictions. Consequently, we collected two additional CRISPR-I screens of E. coli. Figure A shows the distribution difference of lawful change in these three datasets. Figure B shows that models trained on single datasets tended to perform best on their cognate test set. Combining two datasets show better generalization across datasets without degrading performance. And integrating three datasets performed the best. As mentioned before, the strong predictive features for depletion are gene-specific, which is problematic for predicting the accurate guide efficiency. Therefore, we took two approaches to separate guide and gene effects. One is a mixed effect random forest, or MERF. Second, which we refer to as median subtracting, MS. We trained a random forest and a lasso model on MS data and compared to MERF and a published MS method-based lasso model, which we refer to as POSTER. We evaluated the models by calculating the Spearman correlation of all guides for each held-out gene, instead of held-out guides. The median Spearman correlation in tenfold cross-validation is shown in the table. In sum, random forests trained on multiple datasets outperform simpler regression models. To understand the features underlying model performance, we again examine sharp values for our random forest models. Interestingly, top features are near the PAM region, as shown also in the summary plot in Figure B. With sharp interaction values, we further observe potential interactions such as between distant features and sequence features, an example shown in Figure C. Next, we validated the model by silencing GFP in a plasmid using a flow cytometry-based assay and reanalyzed the data from a Miller assay previously used to validate the Pasteur model. Both tree-based models outperform tools designed for the Cas9 system in eukaryotes, but the Spearman correlation might be biased because of the small sample size. Therefore, we performed a saturating high-throughput screen targeting 9 genes in purine biosynthesis pathways known to be essential in minimal media. With this fully independent screen, we again show that random forest models perform the best and data integration improved the model performance. Unexpectedly, we also observed a weak correlation between guide efficiency and distance to the stop codon, different from previous findings. To summarize, we developed a predictive model for CRISPRi guide efficiency using integrated data from 3 gene essentiality screens in E. coli. We have shown that our model improves on the previous state of the art, using both gene-wise cross-validation on our training data as well as a fully independent screen. These investigations provide a blueprint for developing similar predictive models. We also built a web server to design guides and predict efficiency given a sequence based on the MERV model. And a Python script is available on GitHub to design guides for multiple sequences. For more details of the project, please visit poster number 178 on July 13 or check out our preprint. Thank you for your attention.